Okay, thank you very much. Wish to thank the organizers as well for this opportunity to present our data and to continue with a theme we started earlier today on the aspect of inflammation and neurodegenerative diseases. These are my disclosures. The overview for today's talk will be in these 4 points. We're going to talk about ALS. For those of you who may not be too familiar with this disease, we'll go through it somewhat briefly. An enzyme called the SOD1, superoxide dismutase, we'll talk briefly about. Viruses in motor neuron disease, along our viral talks that we've had already, is really what we're going to end with, and one in particular called the chlorovirus, one that we've been doing research the last few years on. So when we're talking about ALS, we ask, what exactly is this? ALS, amyotrophy, lack of muscle trophism, lack of muscle growth, wasting of muscle tissue. That's what we'll see. Lateral has to do with corticospinal tracts within this thoracic spinal cord section. And I think we can kind of show, oops. Pardon me, let's see if my pointer works. These are the corticospinal tracts laterally on both sides, all right? And these are the tracts that become affected, as are the anterior horn cells on both sides. So we get wasting of both of these elements. Neuropathologically, there's really nothing else that looks quite like this. It's a progressive wasting disorder. The hallmark clinically is a painless progressive motor weakness that we'll see in the spinal onset cases. Normally begins with mild symptoms such as weakness, one or more muscle groups, gradually affects most skeletal muscle. It actually proceeds, or it progresses in a segmental fashion. So a patient doesn't present with a right hand weakness and then they have a left foot drop. It would be a progressive process. That's where it's kind of gotten into the whole concept of prionopathy as a mode of spread, which we won't discuss, but it is unique in that fashion. It's generalized, it's not restricted to one body region, it may randomly affect any body, part of the body. 25% it's bulbar, which is the area of my research for the most part. Spinal onset, 75%, very rare respiratory, very rare patients will present with respiratory symptoms as they're presenting symptoms. Unfortunately, this is a fatal disease, and that's why we're working hard to see what changes can come about. Now, here's just somewhat of an idea of what we're talking about. You see the finger contractures, flexor contractures of the fingers, the wasting of the interossei dorsally and the first DI dorsally. On the palmar aspect, the thenar and hypothenar MNI, the volar interossei are affected, and of course, all the flexure deformities. Now, this is the hand you could see potentially with a neuropathy, severe CMT, but when you put everything else together, and if this were a motion picture, you might see fine fasciculations through these thin thigh muscle groups. But this is all what we will typically encounter with patients with this disease. So it's a wide spectrum of pathologic findings, suggesting a syndrome rather than an individual disease state. We see motor neuron atrophy, typically within the brainstem, affecting bulbar musculature, spinal cord affecting the arms and legs, or motor neuron loss. This is a schematic, not to spend a lot of time on, but it just kind of walks through how we start from the top of the motor cortex within the parietal bet cells, and as they course their way along the cortical spinal tract, through the medulla into the crossing of the pyramids, down into the spinal cord, they break off either into the arm muscles, for example, or in the brainstem, the bulbar musculature. And we have what we call lower and upper motor neuron findings, dictated whether it's in the periphery versus in the central region. And not to belabor these points, but I just mentioned it because this is very unique to this disorder. If this tongue were moving, it not only would be atrophied, wasted, but it would be fasciculating. It would be twitching. If anybody has seen this disease, you can't escape it. One of the things we oftentimes see is this yellow debris over the tongue. It's leukoplakia. It's a buildup of bacteria on the tongue, which can foster bacteria. And when they aspirate their spit, saliva can lead to pneumonia. So something we always focus on. But I wanted you just to kind of see, this is a picture, sort of 1,000 words. You don't really see anything quite like this, the disease process. Now, I apologize for the clarity of the slide. These just point out some of the risk factors predisposing to ALS. There are genetic risk factors. We won't talk on all of them. We'll focus on the SOD1 today. There's the TDP43 and RNA binding protein, C9orf72. It's a hexanucleotide repeat, probably the most common form of a gene-induced ALS, at least in America. And TBK1, interestingly, it not only presents with ALS, but FTD. Environmental factors, still looking at the list, exposure to heavy metals has become very popular. Pesticides. I'm from a very rural area, Nebraska. We see, in fact, interestingly, it's the #1 state in the country for Parkinson's. And so why is that? Well, it may actually be the pesticide nature or something within the underwater aquifer water supply. Exposure to electromagnetic fields, and then BMI and nutritional status have also been postulated. Here's an older actually graph from Bob Brown's data. This dates back several years, but it just shows the growing evidence of genetic findings leading to this particular disease. Protein homeostasis, altered RNA binding protein, cytoskeletal protein involvement. About 10–15% of the disease is gene-based. That may change as we learn more about the genes. And here's a simple slide, right? Actually, I just wanted to point this out because this is not a simple disease, or we would have had ways of treating it by now. This is a very complicated disorder. And if you look at the cell body of the motor neuron and the axon, you see a number of different mechanisms affecting the cell that actually we find a significant effect on. And let's just look at the one with SOD1, retrograde axonal transport, non-cell autonomy, the astrocytes, microglia, oligodendrocyte, SOD1 influences, endoplasmic reticulum stress, mitochondrial dysfunction, which studies have shown may be some of the earliest organelles to be affected with this disease, as well as anterograde axonal transport, protein aggregation. So this one particular enzyme, if defective, could have multiple effects on the cell and the disease mechanism itself. So what is this SOD1? Well, SOD1 is called superoxide dismutase. There are 3 forms. We focus on the first form, or the #1. In 1993, a large collaborative effort demonstrated certain inherited forms of ALS. This was Bob Brown, some other people. This is the first gene that was identified as causing ALS. It's a genetic change altering an abundant enzyme within the cells called the copper zinc superoxide dismutase as an enzyme. And it's abundant throughout the cells, of course. SOD1 functions by catalyzing the dismutation of 2 superoxide radicals, hydrogen peroxide and O2, has a copper ion held within the enzyme assisting with the disulfide bonds, and a zinc atom maintaining enzyme structure. To date, over 200 different mutations in this enzyme have been identified. Now, just to turn a little bit onto viruses, these infectious agents such as viruses have been postulated as contributors to neurodegenerative diseases, motor neuron disease in particular. And we've seen this with West Nile 15 years ago. We had reported cases of motor neuron. Polio as a lower motor neuron. HIV, we've had multiple cases of HIV cases actually being treated and sometimes even seeing the motor neuron improve. The cyanobacteria, beta-methylaminoalanine is found in green-blue algal blooms, and it's been known to actually lead to a motor neuron syndrome. And a human endovirus, it's a retrovirus, it's called HERV-K. It's been found to be part of the human virome and also tied in with potential for ALS. A colleague of mine at Duke, Rick Bedlack, reported COVID-19 accelerating disease progression in 2 patients. So this does not say causing ALS, just says individuals with disease getting worse. I'll come back to that point as we get into chloroviruses and motor neuron disease. So most of us probably have never heard of chlorovirus. Certainly I hadn't before we got involved in this. But chloroviruses have been around for a very long time. They represent an environmental exposure only recently associated with certain human pathogenic conditions. They're large, very large, in fact, double-stranded DNA viruses, ubiquitously found within freshwater environments worldwide, first identified actually in Germany. They infect and replicate in specific green algal blooms, and I'll show a picture of that in a second. Chloroviral virions have been shown to infect mammalian macrophages, which is a focus of what we'll be discussing today, in vitro, inducing inflammatory cytokines, while infection of mice has induced neurocognitive deficits. So This is sort of what it looks like under EM. You see these green algal blooms with these little chloroviral attachments. They seem to come out in the spring and in the fall on freshwater lakes and reservoirs. And I find it interesting only because we've had numerous cases of individuals with ALS living in proximity to these freshwater reservoirs. Got one town of 23,000 people in Nebraska who we have 3 cases within the east to southeast corner of this lake. And this is a disease you should have 1 to 2 per 100,000. So we're fairly convinced there may be something going on. It's yet to be proven. So just to dive into this a bit, the Acanthus cystis cerevisiae chlorovirus, big name, we'll just refer to as ATCV1, is a member of one of the 4 known chloroviral types. But what makes it interesting is it encodes its own functional copper zinc superoxidase mutase. Almost no other viruses do this. This is very unique, and that's what got me as a clinician interested in looking into this. Now, the ATCV1 SOD1 is predicted to lower the concentration of reactive oxygen species in cells early during viral infection, thereby evading the innate antiviral resistance. So it's a self-protective mechanism for the virus. Obviously, of clinical significance, as we've talked about the SOD1 variants with ALS. So the importance of the abnormal SOD1 in ALS is also represented in sporadic ALS. And what I mean by that are individuals with the disease who don't have a genetic connection, which makes up 85, maybe 90%. Because we have found evidence of SOD1 dysfunction in these individuals as well, leading us to the hypothesis that ATCV1 SOD1 may influence the pathogenesis of ALS. So the hypothesized pathogenic effect of ATCV1 on humans is relatively recent. It was thought to only infect certain eukaryotic flora like green algae, which are endosymbionts of the heliozoan Acanthus cystis cerevisiae, a common protost of inland waters. However, infectious CVs are found at times with major spikes, and that's the one point that we've been very interested in. Are we seeing a higher incidence of disease when we see higher incidence of spikes of this particular virus? As mentioned, there are 4 basic groups, 43 total isolates. Not all aquatic systems have all these isolates, but there seems to be a high chance of human exposure to these chloroviruses. Furthermore, it appears that exposure to chlorovirus is highly significant following the recent detection of chloroviral DNA sequences in the human virome. So we're stumbling across something that likely could have been pathogenic for some time. So we decided to do some research, and our research has found that mouse macrophages support low levels of the ATCV1 replication in vitro. This also was accompanied by production of inflammatory cytokines. Therefore, plausible that persistence of the chlorovirus within the macrophage stimulates the production of immune factors contributing to neurodegenerative diseases such as nitric oxide, interleukin-6, interferon-stimulated genes, almost serving as a Trojan horse, carrying these mechanisms, inflammatory maybe, throughout the body, potentially through the blood-brain barrier. So we hypothesize that the ATCV1 persistence in macrophages inducing the inflammatory cytokine production could be a mechanism by which these viruses could impair neurological function. So this hypothesis falls on a corollary of the well-established theory that high levels of pro-inflammation impair not only cognition, but behavior and neurological function, leading us to our second hypothesis that this virus may play a role with ALS. So off we went. We looked at 2 different types of studies, just to briefly run through this. We took samples from individuals, from humans with the disease and controls, and we looked for antibodies to the chlorovirus. And we also did something with transgenic SOD animals. We had a SOD1 G93A transgenic mouse model, and we also had a wild type mouse model that we either injected intracranially with the virus, with saline, or with something called poly-IC, which induces an innate antiviral immune response. So 2 separate processes going on at once. The human data, what we found is that of the 17 ALS patients that we just took blood from, just normal ALS patients, so to speak, they all had significantly high levels of IgG1, which is the antibody to the chlorovirus. The 13 controls also had levels, but they were very low. Statistical difference between the 2, suggesting everybody's been exposed. But why are the ALS patients having a higher titer to this particular virus, something that we really didn't have a great answer. Now, the IgG1 is the most abundant IgG subclass, mediating antibody response against viruses. Maybe that's why that was up. But ALS patients would appear to have either had a significantly greater exposure or a greater antibody response to the virus. And here we can see this particular difference between our control group and our ALS patients reaching significance. Now, in the mouse model, ATCB1 infection significantly accelerated the onset and progression of motor neuron disease in these transgenic mice. These mice only live about 155, 160 days. That's about their life cycle. And we saw that by about 140 days, we started seeing the animals injected with the virus getting sick fast and going downhill quicker than their litter mates who were not injected. Now, accelerated mortality was not identified, but interestingly, treatment with the poly-IC resulted in a significant delay in mortality in the transgenic mice compared with those receiving saline. So I don't know if you've had much exposure to mice or what they look like, but the focus has to do with their limb integrity. It has to do with the hind limbs in particular, the forelimbs, holding the rotor rod. There's a number of different measures that we use, but this is sort of an end stage of what these animals appeared to look like. And here's a progression curve where, simply put, the black and red dots are the two different types of chlorovirus, ATCV and PBCD and others that we use, showing the onset and progression faster than the saline in blue. Yet, they all sort of converged at the end. It didn't really, as the next slide, the KM slide showed, we didn't really see a significant change in survival amongst these groups. So it seems to propagate the disease, and it seems to do so in animals that are genetically susceptible, because in the wild type animals who can live 2 years, these animals that we injected lived 2 years. There was actually no effect at all. So this really gets down to that whole concept of if, like you had mentioned earlier, if somebody's prone to having dementia, does COVID potentially jumpstart it? Well, we're kind of thinking the same with this particular virus, with this disease. So we feel that this appears to confirm that this viral infection accelerates motor deterioration, at least in the SOD1 transgenic mice. Now, in addition to the SOD1 progression, the immune system factors, such as IL-6, ISG, are associated with ALS in humans and MND in these transgenic mice. We had showed previously that inoculation of the mice led to persistence of viral nucleic acid in the brain, higher than normal inflammatory factors, interferon gamma, macrophage microglial markers, subsequently concluding that the ATCB1 infects macrophages, inducing these inflammatory responses, and it also encodes its own SOD1. And here we can see the gamma interferon graph to the left and the IL-6 to the right, both showing significant elevations in our ALS patient population. So in summary, the ATCB1 SOD1 seems to augment production of inflammatory factors associated with the development of ALS in both humans and transgenic mouse models. So to date, we feel we've identified a cohort of ALS patients with significantly elevated IgG1 antibody compared to control. We've seen the degradation of motor performance in those animals, transgenics treated with the virus, and the expression of the ATCB1 SOD1 in responding mouse macrophages resulted in enhancement of inflammatory mediators and ISG promoter activity. Now, given that treatment with the poly-IC delayed the onset of motor neuron disease, it's plausible that active chloroviral infection or a defective component of the antiviral immune response against active viral infection is promoting early onset motor neuron disease. All plausible, possible. These are our theories, basically. Questions? Several, actually. What are the mechanisms by which humans are exposed? We actually checked Lincoln water supply, and we found a significant amount of chlorovirus in the water, but we didn't know it was heat killed or live. We couldn't tell, but it was interesting. It seemed to be pretty ubiquitous. Could respiratory inoculation or oral ingestion prove to be the means of exposure? Of course, ours was with the intracerebral injections. Given that measurable serum antibody titers against this virus in humans have been detected, a common means of exposure would be suggested. And is there transit through the blood-brain barrier, which may involve viral-induced immune mechanisms targeting the motor system, all of which need to be answered. future directions focusing on some of these questions, looking at other types of transgenic mice to see if they also have an effect from their genetic abnormalities, or the contribution of other chloroviral types on other transgenic animal models. The alternative routes of inoculation we talked about, the timing of inoculation, earlier, late in the life cycle, prior to the disease onset, all seem to be playing a role. Direct infections of neuronal cell lines involving SOD1 G93 overexpression are currently under investigation in our lab to further understand if and how these viruses contribute to motor neuron disease through a cell-specific pathogenesis. We need to further identify exactly how humans are routinely exposed, as I've already mentioned. To what degree does it cross the blood-brain barrier? Does exposure to the viruses early in life have an effect? What about the gut microbiome? We've seen a lot in some of the slides earlier today with the vagus nerve innervating the gut. Could that be a means of getting the virus into the CNS, and is that actually playing a role? Ultimately, it's essential to determine the roles and involvement of macrophages in the viral-induced motor neuron disorder. So in conclusion, we feel that the exposure to this virus appears to accelerate the onset of an ALS-like motor neuron disease disorder in transgenic mouse model, and that the viral SOD1, unique to this virus, may augment the induction of inflammatory factors from macrophages, and suggesting in summary that this may be contributing to pathogenesis of this disease process. So those interested, we have an article in the Frontiers in Neurology from a couple of years ago that goes through this in a little bit more detail. These are my collaborators. The work was supported by the Stuart Nichols ALS Research Foundation. And I thank you for your attention.
Thank you.
Any questions?
Yeah. First of all, thank you for a really well done, very thought-provoking talk. The question that immediately came to mind, that Florida is these days eaten alive with blue-green algae blooms, in good part due to the problem that ways of the state that we use around fertilizer runoff in the water supplies and render food bodies of water just can't go there. But partly due to climate change. So yeah, we've got a real green algae crisis in Florida. Yeah. So I wondered if there were quantity data on motor neuron disease that suggested that if you live in a place like Florida, that the disease tends to progress more rapidly.
That's an excellent question. Actually, I don't know if you know Walter Bradley. He's a sort of a renowned neurologist in ALS. He's done a lot of work on the blue-green algae in Florida, having lived there. Gets back to that one earlier slide with the BMAA, the cyanobacteria, and the effect that this may have. They've done a lot of work. They've got a lab out in Jackson Hole, I think, Wyoming, where they're focusing on the effects of this excitotoxin in motor neuron disease. There's a Guamanian form of ALS, which has a similar excitotoxin, which has also been contributing not only to ALS, but dementia and Parkinson's. So we're really very focused now on these potential infectious exposures, toxic exposures. People in Michigan are doing a lot of really good work looking at metals, lead exposure. They've actually done some interesting data looking at wisdom teeth of individuals with ALS. Wisdom teeth are pulled in their teens, get the disease in their 50s. They go back and they analyze the wisdom teeth, high levels of metal in the wisdom teeth. Interesting, suggesting could there have been an earlier exposure? But no, your point is very well taken, and that's something that we've also, of course, been very interested in. And they've done studies looking at communities downstream. You know, winds across North America go from west to east, of course, and they're finding in villages and little townships east of large reservoirs higher incidence of ALS than what we would see on the west side. It's been pretty well documented, actually. So there must be something to it. What makes this such a tough one is that, I think, as you had mentioned before, nothing happens in a vacuum. I mean, these disorders are going on with so many other associated comorbidities that you can't always tease apart just one mechanism. Keeping you guys from lunch, sorry.
So, the last question maybe. Did you
you know the levels of ISOD1, 2, or 3 in peripheral area compared to central nervous system.
So we didn't look at SOD2 or 3. No, we
It's
just
only
focused,
one.
just focus on
You have
SOD1.
an idea about the level in peripheral compared, specifically in spinal cord,
Yeah.
compared to the central nervous system.
Yeah, no, problem twofold, actually, because the transgenic mouse model is designed that way, and also because the particular viral, the variant had the SOD1. One thing that we didn't do yet, which would be very interesting, is to see the virus within the tissue. I mean, almost like, are we seeing COVID within the tissue? Is it an inflammatory cascade that's accelerating these processes? Maybe in a genetically predisposed organism, you give a viral insult, and that may trigger the mechanism to get started. These are some of our postulates that we've been wrestling with. Yeah.
Yeah, we need to look to the.
Okay, thank you very much. Wish to thank the organizers as well for this opportunity to present our data and to continue with a theme we started earlier today on the aspect of inflammation and neurodegenerative diseases. These are my disclosures. The overview for today's talk will be in these 4 points. We're going to talk about ALS. For those of you who may not be too familiar with this disease, we'll go through it somewhat briefly. An enzyme called the SOD1, superoxide dismutase, we'll talk briefly about. Viruses in motor neuron disease, along our viral talks that we've had already, is really what we're going to end with, and one in particular called the chlorovirus, one that we've been doing research the last few years on. So when we're talking about ALS, we ask, what exactly is this? ALS, amyotrophy, lack of muscle trophism, lack of muscle growth, wasting of muscle tissue. That's what we'll see. Lateral has to do with cortical spinal tracts within this thoracic spinal cord section. And I think we can kind of show, oops. Pardon me, let's see if my pointer works. These are the cortical spinal tracts laterally on both sides. All right. And these are the tracts that become affected, as are the anterior horn cells on both sides. So we get wasting of both of these elements. Neuropathologically, there's really nothing else that looks quite like this. It's a progressive wasting disorder. The hallmark clinically is a painless progressive motor weakness that we'll see in the spinal onset cases. Normally begins with mild symptoms such as weakness, one or more muscle groups, gradually affects most skeletal muscle. It actually proceeds, or it progresses in a segmental fashion. So a patient doesn't present with a right hand weakness and then they have a left foot drop. It would be a progressive process. That's where it's kind of gotten into the whole concept of prionopathy as a mode of spread, which we won't discuss, but it is unique in that fashion. It's generalized, it's not restricted to one body region, it may randomly affect any body, part of the body. 25% it's bulbar, which is the area of my research for the most part. Spinal onset, 75%, very rare respiratory, very rare patients will present with respiratory symptoms as they're presenting symptoms. Unfortunately, this is a fatal disease, and that's why we're working hard to see what changes can come about. Now, here's just somewhat of an idea of what we're talking about. You see the finger contractures, flexor contractures of the fingers, the wasting of the interossei dorsally and the first DI dorsally. On the palmar aspect, the thenar and hypothenar MNI, the volar interossei are affected, and of course, all the flexure deformities. Now, this is the hand you could see potentially with a neuropathy, severe CMT, but when you put everything else together, and if this were a motion picture, you might see fine fasciculations through these thin thigh muscle groups. But this is all what we will typically encounter with patients with this disease. So it's a wide spectrum of pathologic findings, suggesting a syndrome rather than an individual disease state. We see motor neuron atrophy, typically within the brainstem, affecting bulbar musculature, spinal cord affecting the arms and legs, or motor neuron loss. This is a schematic, not to spend a lot of time on, but it just kind of walks through